Good afternoon, students. Welcome to the class. Now, today we will continue our chapter. And yesterday I gave you some basis for the Mendelian inheritance. Now, today we'll discuss Mendel's laws of inheritance. Law of dominance, law of segregation, and law of independent assortment. Now, law of dominance, according to this law, hybrid offsprings will only inherit the dominant trait in the phenotype. The alleles that are suppressed are known as recessive traits, whereas the alleles that determine the trait are known as the dominant trait. I hope you have gone through the terms and now you are familiar with these terms: phenotype, genotype, trait. Phenotype is the expression which you can see, and the genotype is the genetic composition or genetic makeup. Then law of segregation. Law of segregation states that during the production of gametes, two copies of each hereditary factor segregate so that the offsprings they acquire one factor from each parent. In other words, alleles, that is the alternative form of the genes, pairs these segregate during the formation of gamete and reunite randomly during fertilization. Law of independent assortment. It states that a pair of trait segregates independently from another pair during gamete formation. As the individual hereditary factors they sort independently, different traits get equal opportunity to occur together. We will take all these one by one. We will start with the law of dominance. I will try to explain it with an example first. We will take the example of cherries. Now, how we can explain it? Let's now pretend that the cherries they carry gene X for color. This is our assumption, right? So each cherry has two copies of gene X, and those copies are known as alleles. We can say capital A and small a. So capital A codes for the red color and the small a codes for yellow color. Now, this law of dominance, it says that when an organism is heterozygous for a trait, then only the dominant allele will produce the phenotype. That is why you can see here in this figure which is displayed on your screens that the first cherry is homozygous for both the alleles, that is it has capital A and capital A. Second cherry is homozygous for the yellow allele, it has small a and small a. And the third cherry is heterozygous, that is it has one allele for the red and one for the yellow, one capital A, one small a. So this cherry phenotypically appears red. So the law of dominance says that the allele A, capital A, that is the red one, is dominant because it is producing the phenotype in a heterozygous organism. So from here I hope it's clear to you that when an expression is expressed in the heterozygous condition, it masks the expression of the other allele, then that particular allele is known as dominant allele. So now we come down to the Mendel's monohybrid cross. Now what do we understand by monohybrid cross? That is the inheritance of one gene. First, I'll talk to you about it in general. So, Mendel, studied the inheritance of a single character at a time. Obviously, the plant was as a whole, but the focus was on a single character. So, the breeding experiments dealing with the single character, we call them as monohybrid cross. And in these experiments, only one character or trait is at focus. So Mendel considered the size of the stem. For example, we'll take here like it is displayed on your screens. Okay. So on your screens you can see this. Now you can see in red it is written F1 generation. Above F1 is written the tall and the dwarf, that is the parental generation, which is written the tall and short or the dwarf. Capital T and capital T are the two alleles which are representing the tall plant, and small t and small t are those which are representing the dwarf plant. So he cross-pollinated a pure tall pea plant with a pure dwarf pea plant. And the plants which he took from where he started his experiment, he called them as parental generation. So nowadays we represent it by the symbol of P, the parental generation. He collected the seeds produced by this crop and grew the plants from them in the next season. All these plants were found to be tall, as you can see where it is written F1. There was no short plant. So they constituted the F1 generation, that is the first filial generation. So results were same whether female parent was tall or male parent was tall. So reciprocal crosses, that is, they gave the same result. Now these F1 plants, they were pollinated amongst themselves, that is what is known as selfing or inbreeding. The flowers were kept covered from the beginning with paper bags to avoid contamination with any unwanted pollen, so that the experiment may not go waste, and the seeds produced by all the F1 plants were again collected and then again sown in the next season. These plants constituted the F2 generation or the second filial generation. The F2 plants were found to contain both the tall as well as the short plants or the dwarf plants, and they were found to be in a ratio of 3:1. 3 tall to 1 dwarf. That was the ratio. It's not that only 4 plants were formed. This is the ratio of a number of plants which were sown. So, the genetically mixed offsprings they produced by crossing, they produced two pure parents and having contrasting traits. These are known as hybrids. So, hybrids were formed in the F1 generation, and then by their selfing they formed the pure forms as well as the hybrid forms. Clear? Now, as far as I told you that these crosses give similar result with reciprocal parents also. So what is a reciprocal cross? A reciprocal cross involves the same traits but carries the sexes are opposite to those which were in the first case. For example, if in the original cross, the pollen from the flowers of a tall pea plant were transferred to stigmas of flower of a dwarf or a short plant. In reciprocal cross, the pollen from flowers of dwarf plant will be deposited on the stigma of the flowers of the tall plant. That is, the characters by purity will be same, but the male and female parent can be changed, but the results are same. So what were the findings of or the results of this monohybrid cross? So Mendel got the following results, that is, firstly, F1 plants produced by a cross between two plants pure for the contrasting traits of a character are similar and express only one contrasting trait. That is, they resemble only one parent. No intermediate between the two alternative traits of the parents. Secondly, it does not matter which parent provides the male gamete and which provides the female gamete. The results remain the same. That is, the reciprocal crosses produce same F1 plants. Then both the contrasting traits, they appear in the F2 generation. The contrasting trait of a character expressed in F1 plants appears in 3/4 of the F2 plants. One of the two contrasting traits of a character remains unexpressed, that is hidden in the F1 generation. As you can see here that in F1 generation there is no short plant. So that is hidden trait. In F1 no short or dwarf plant means the hidden trait of the shortness or the dwarf plant. So what are the conclusions which can be drawn from this monohybrid crosses in general? That gametes bring from the parent something which makes a character appear in the next generation. This something was known as factor by Mendel. The inherited characters are thus determined by separate unit factors. And there are a pair of unit factors for each character, one inherited from each parent. For example, height in pea plant is controlled by two factors, since some plants are tall and some are short or dwarf. So now we'll take this example in another way and I'll show you here how we do it. This is the way we represent it. So we'll take the example of this hybridization experiment which was carried out by Mendel between tall and the dwarf pea plant. To study inheritance of one gene, that is the height. What he did was he collected the seeds produced as a result of this crop and grew them in to generate the plants of the first hybrid generation, known as the first filial generation. As I just told you, the parental plants on your screens, you can say they have these two alleles, the capital T, and the dwarf one is represented by small t and small t. Obviously, they will produce the gametes of one time only because gametes are produced as a result of meiosis. So, only one of the alleles will be present in each gamete. Now, here one is shown because it is of the same type, similar are produced. Why? Because the parents are having purity in the character. Both the alleles are same. Now F1 generation or the first filial generation, all the plants were tall, right? But none were dwarf like one of its parents. So this means that F1 resembles either of the parent and that the trait of the other parent is not seen. And the trait which represents itself, then we call it as dominant trait. Mendel then self-pollinated these F1 tall plants, and then to his surprise he found that in the F2 generation some of the dwarf again appeared. The character which was not seen in F1 again expressed itself in F2. Now how they appear here in the F2? Can you tell me? I hope you have understood it by now. Now this F2 plants, we can understand it by the diagram which is displayed on your screens, which is also known as the Punnett square. Now this Punnett square was developed by a British geneticist known as Reginald C. Punnett. So it is after the name of this scientist that, or a genetist to be more precise, we call it as Punnett square. This is a graphical representation to calculate the probability of all the possible genotypes of offsprings in a genetic cross. The possible gametes are written on two sides, as you can see, usually on the top row and the left column. All possible combinations are represented in boxes below in the squares, and this generates a square output form. The Punnett square shows the parental tall, that is the pure tall, capital T and capital T male, and the dwarf small t and the small t. Right? So the gametes which were produced here in the F1 generation are capital T and small t, and then on self-pollination they produced the F2 generation in the Punnett square. So here you see that what happens is that you can see out of the 4 types which are formed, one is capital T and capital T, that is the pure tall. That means that 1/4 of the random fertilizations they lead to capital T and capital T. Then in 2 columns you can see capital T and small t. This means that 1/2 will lead to the formation of hybrids, hybrid tall like F1 generation, and in only 1 column you see small t and small t, which is appearing or rather reappearing in this generation. That is 1/4 will form the small t and small t. Although in the F1 we see that the genotype is capital T and small t, but the phenotypic character which was seen was tall, whereas in the F2 we see that 3/4 of the progeny, that is the capital T1 and the capital T and the small t1 were tall, whereas the small t and small t, only 1/4 of it was dwarf. So here if we see the phenotypic ratio of tall and dwarf in the F2 generation, it comes out to be 3:1, right? But if we look at its genotype, the genotypic ratio is 1 tall is to 2 hybrid tall, that is 1 pure tall is to 2 hybrid tall is to 1 dwarf. That means 3 types in the genotype, 1:2:1. But what you see or visualize as the expression, that is only 2 types, tall and dwarf, And if you consider the 4 blocks here as the 4 different parts, then you can see the 3/4 part as tall and only 1/4 part as dwarf. That is 3:1 is the ratio in which they were formed. Okay. Now if we further continue to F3 generation, then self-pollination of the dwarf ones will lead to the formation of only dwarf plants, whereas the self-pollination of the hybrid talls and the tall, that is in general if we say the tall plants, will lead to the production of again all the types, that is pure tall, hybrid tall and the dwarf plants. I hope you got it. I made it clear. So according to the law of dominance, the characters, they are controlled by discrete units which are known as factors, and these factors, they occur in pairs, and in a dissimilar pair of factors, one member of the pair dominates the other, and the one which dominates is known as the dominant, and the one which does not express itself and is suppressed in the presence of the dominant one is known as recessive. Right? Now this is according to the complete dominance and recessiveness. But supposing the complete dominance and recessiveness does not occur, then what will happen? This means that there is incomplete dominance. Now if you have got the concept of monohybrid cross with the dominant and the recessive part clear, then we can proceed on to the next part, that is the incomplete dominance, which is also known as the blended inheritance, although the characters don't actually blend. Right? What we saw in the experiment just now, which we studied, that is the Mendel's P experiment, that dominance was essentially complete. There was no difference between the homozygous and the heterozygous plant in the expression of a dominant character phenotypically. Right? But this is not true of all characters of organisms. There are characters or alleles that are not dominant or recessive exactly in black and white or clear cut manner. In such cases, both the alleles of the contrasting conditions of a character can express themselves as a blend. With the result, the hybrid produced by the crossing of two pure individuals does not resemble either of the parent, but is somewhat midway between the two parents. So, the expression of the traits of two pure parents as an intermediate condition or fine mixture in the F1 hybrids is known as incomplete or partial dominance. This is also known as blended or intermediate or mosaic inheritance. This is due to the fact that the dominant character or the gene is not in a position to completely suppress the recessive one, with the result that the heterozygous has a different phenotype, which is different from the homozygotes of either of the allele. Right? This is exception to the Mendel's principle of dominance. Now the examples of this we can take as very good examples which are in the plant kingdom, that is of Mirabilis jalapa, 4 o'clock plant, which is displayed on your screens, and Antirrhinum majus, that is the snapdragon, and in the animals, the Andalusian fowl. So we'll take one example of 4 o'clock plant, that is Mirabilis jalapa, which is displayed on your screens right now. So this is a cross between a plant pure for red flowers, you see the color here, and a plant pure for white flowers. So the focus gene here is the color red and white. But when they are crossed, they yield hybrid plants with pink flowers in the F1 generation. Normally this is not expected. Red and white flowers, they give rise to pink. Okay, so F1 generation is all pink, neither red nor white. So this means both are not completely dominant. So both these colors, they appear and blend the color into pink one. Now this is a contradiction of the Mendel's assumption that no blending of characters takes place. But you see, when two of the hybrid plants with pink flowers are crossed, the F2 generation includes plants again with red, pink and white flowers in the usual Mendelian ratio of 1:2:1. How come? We'll see now. So this is according to the Mendelian ratio, but the thing is, the characters, they did not infuse with each other. They are again segregated and they again appear in the F2 generation. So this cross, it shows incomplete dominance. The genes for red and white color do not actually mix with the F1 generation to form the pink hybrids, as both the characters, that is red and the white, they reappear in the F2 plants. There is no specific gene for pink color. Make sure. Genes are only for red color and white color. No specific gene for pink color. Quantitative effect of the gene comes the homozygous plants, they are unable to produce the flower pigment, and the heterozygous, they can, right? So there are these different pigments, the concentration of the pigment which gives the color. Here we represented in the parental generation the red color as capital R and capital R because it is homozygous, and the white as the W and W. We can also represent it as small r to be more easy. So here we take it as R and W. So here you see the resultant capital R, capital W of the F1 generation are pink because they can produce only half the amount of the flower pigment which is produced by the capital R and capital R homozygous alleles, right? So in the F2 generation, the genotypic ratio is the same as the Mendelian ratio, that is 1:2:1. 1 is capital R and capital R, 2 parts is capital R and W, whereas 1 part is W and W. So 1 part is red, 1 is white and 2 are pink. So here the phenotypic ratio and the genotypic ratio is the same. So phenotypic ratio is also 1:2:1 and the genotypic ratio is also 1:2:1. So this ratio 1:2:1 in the phenotype of F2 is a characteristic or the diagnostic ratio of incomplete dominance. I hope it's clear. If any doubts, you can just ping me. Again, I'll explain you a little about the incomplete dominance again, if any doubts are there. You see here neither of the allele is dominant. You can see three different colors of flowers on your screen: red, yellow, and intermediate, the orange type. So when an organism is heterozygous for a trait, it will show a third type of phenotype, right? So what here you see, there is one red flower, right? Here this A, capital A letter represents the gene, and R and Y represent the different alleles. So capital A with R, if both the alleles are like this, the flower is going to be red. And if it is capital A with Y, then it is going to be yellow. But in the hybrid of both these, we get one capital R A and one A with Y. So A with R and A with Y. So here the color is intermediate between the two flowers. It is neither red, it is nor yellow. So this is what is incomplete dominance. Although in the F2 they may be again represented. Now one more thing which we are going to discuss today before we end this class is about the back cross and the test cross. So now what is a back cross and what is a test cross? That is what we will see now. I hope incomplete dominance is clear to you. We have studied the monohybrid cross, then the incomplete dominance, and now we are going to study the test cross. This diagram which is displayed on your screens is also in your textbooks, and here you can see both the back cross as well as the test cross, how we can determine the genotype of a plant. Here it is written the capital P and capital P and small p. That means whether if this flower, color of the flower, that is the violet color of the flower, we have to check whether it is homozygous or heterozygous. What should we do? It is, back crossed this with the parental recessive type. So here the homozygous recessive is the white flower. So there are two types of possibilities, alternative 1 and alternative 2. So all the offsprings, they can be purple, that is 100% purple progeny will prove that the purple flower in question was homozygous, that is homozygous dominant. Whereas if there are half the progeny is purple and half is white, that is 50-50 or 1:1, then we can say that it is what? Heterozygous. Now this heterozygosity or this 1:1 ratio shows that it is a test cross for determining the heterozygosity of the flower or the whichever organism we took into consideration. In a monohybrid cross, the this one character, that is the test cross ratio, is always 1:1, but if we take two characters into consideration, then it is 1:1 is to 1 is to 1. So this ratio shows a test cross, and normally the F1 generation, when crossed with the parental recessive type, is what is known as a test cross. And back cross is with either of the parent, that is back cross means with the either the dominant parent F1 when crossed either with the dominant one or with the recessive one. Both are back crosses, but out of the two, only the one with the recessive parent is a test cross. So this shows the purpose of the test cross. Now what is the purpose? That is, we have to show the whether the character in question, where the question mark is put, is dominant, homozygous dominant or heterozygous dominant. So again I repeat, a cross between an individual of unknown genotype and an individual which is homozygous for recessive characters is known as a test cross, whereas a genetic cross between a hybrid organism and one of the original parental types is called a back cross. That means original parental types means it can be either parent, dominant or recessive, because parents carry pure characters either for the dominant or for the recessive trait. So the use for the test cross is to reveal the genotype of an organism displaying the dominant phenotype, right? So this is an important tool for the geneticists to come to derive or come to a conclusion to know whether the character or trait in question is genotypically homozygous or heterozygous. And I repeat again that the test cross is of two types, monohybrid and dihybrid. A monohybrid test cross deals with single character giving F1 progeny of 1:1 ratio, whereas a dihybrid test cross deals with two traits giving a ratio of 1:1:1:1 in F1 generation for a cross between a double heterozygote and a double recessive homozygote, right? So here you can see that if all the progenies in the cross which is shown on your screens are purple in the this F1 generation, then the question flower is homozygous, whereas that is homozygous purple. But if it shows half of the progeny as white and half as purple, then it is 1:1 proportion, which means it is a test cross and the questioned flower is a heterozygous. I hope you got everything. In case of any doubts, please contact me. Good afternoon. Stay safe, stay healthy. We'll meet in the next class.